Okay, let's unpack this. Welcome back to the deep dive. Today, we're diving deep, really deep, into some fundamental signal molecules. We're talking about the messengers that run things in your cardiovascular and nervous systems. Think blood pressure, blood flow, memory, even pain.
Exactly. And our mission today is, well, total mastery. We want you to really get the science, the biochemistry, and the clinical side of these key players.
And we're looking at two really distinct groups of molecules, right? Based on some pretty core medical pharmacology research, specifically Chapter 37 focusing on these guys.
That's right. And they couldn't be more different. On one hand, you've got nitric oxide, NOO. It's incredibly simple. A tiny gas, a free radical, the smallest signal molecule we know of.
And on the other?
The vasoactive peptides. These are complex, long chains of amino acids, intricate structures doing, well, equally intricate jobs.
So a simple gas versus complex proteins, understanding how they interact, how they push and pull against each other. That sounds like the core of it.
It absolutely is. That balance is key to understanding so much cardiovascular pharmacology and physiology.
Okay, so let's start with the famous gas then. Nitric oxide, NO.
Right, so NO, it's an oxide of nitrogen, pretty simple chemically. But the crucial thing is it's a free radical. It has an unpaired electron.
And that makes it reactive, right? Chemically speaking.
Highly reactive, which explains its very short lifespan in tissues. And yeah, it's the smallest endogenous signal molecule we know, and uniquely, it's a gas at body temperature.
The discovery story here is pretty amazing, isn't it? Sort of a classic scientific detective story.
Oh, absolutely. It's a defining moment in medicine. For ages, scientists knew certain things relax blood vessels, but the how was a mystery. The real breakthrough started around 1980.
With Furchgott and Zwatsky.
Yes. They were working with acetylcholine, a known vasodilator, and they noticed something, well, simple but profound. And only relax the smooth muscle and blood vessels if the inner lining, the endothelium, was perfectly intact.
If you damage that lining.
The relaxation just didn't happen, gone. Which led them to propose this idea of an endothelium-derived relaxing factor, EDRF.
Some unknown substance released by the endothelium that told the muscle to relax.
Exactly. They knew something was being released, but they had no idea what. Meanwhile, you had Farid Murad's earlier work back in '77.
On nitroglycerin, right. GTN, the angina drug.
Right. Murad showed that GTN worked its magic by releasing nitric oxide. So you had these two separate lines of research, EDRF from the endothelium, NO from drugs like GTN.
And Ignaro connected the dots.
Luis Ignaro in 1987. He definitively proved that EDRF was nitric oxide. These two mysterious factors were one and the same.
Wow. So linking the body's own relaxation signal to the way these drugs worked, that must have been huge.
Absolutely massive. It fundamentally changed how we understood vascular control. Furchgott, Murad, and Ignaro shared the Nobel Prize for it in 1998. A total paradigm shift.
And it cemented NO's role not just in blood vessels, but across systems, nervous system, immune system, a real key messenger.
A fundamental player. Okay, so how do we make this stuff? And maybe more importantly, how do we control it? Because free radicals can be dangerous.
Good point. Let's get into the synthesis.
It's actually a single, quite elegant pathway. The enzyme doing the work is nitric oxide synthase, NOR. It takes a very common amino acid, L-arginine.
Which we usually have plenty of, right?
Generally, yes. L-arginine isn't usually the limiting factor, so NOS converts L-arginine into NO. The real control point is the NNOS enzyme itself.
And it's a demanding enzyme, needs helpers.
Oh, yes. It needs molecular oxygen, it needs NADPH, and then a whole suite of cofactors, things like FMN, FAD, HAM, and this critical one, tetrahydrobiopterin or H4B.
Okay, don't need to memorize the whole list maybe, but the point is, NOS is complex. It's regulated. Things can go wrong with its function in disease.
Precisely. And that complexity is really highlighted by the fact that we have 3 different versions or isoforms of NOS, each with its own job and its own control switch.
Right. So the first types are the constitutive ones.
Correct. Endothelial NOS, or eNOS, sometimes called NO3, found in blood vessel linings, and neuronal NOS, NOS1, found in nerves.
And constitutive means they're just always there working at a low level.
Pretty much, synthesized at a relatively steady low rate, and their activity is switched on and off very tightly. It absolutely depends on calcium and calmodulin binding together.
So things that briefly raise calcium inside the cell, like acetylcholine or another molecule called bradykinin, you flip the switch?
Exactly. A quick pulse of calcium, NOS turns on, produces a puff of NO, then turns off again. It allows for really fine-tuned, rapid local control. Think moment-to-moment adjustments in blood vessel tone.
Okay, so that's enos and NOS. Precise, regulated. What's the third one?
The third one is the game changer, inducible NOS, or iNOS, also called aNOS2. You find this one mainly in immune cells, macrophages, monocytes, also liver cells.
And inducible means it's not always around.
Correct. It's normally present at very low levels, but its production gets massively ramped up, induced by signals of infection or inflammation. Things like bacterial toxins, cytokines like TNF-alpha.
Okay, so as part of the immune response, what's the key difference in how it's controlled?
This is absolutely critical. Once iNOS protein is made, once it's induced, it does not need that calcium calmodulin switch. It's basically active as soon as it's synthesized.
Whoa! So no fine-tuning there.
None. It just churns out NO. And because it's induced by inflammation, it produces absolutely huge amounts of NO, orders of magnitude more than eNOS or NOS, and it can keep going for hours, even days.
So eNOS NOS are like precision regulators, tiny adjustments. iNOS is like opening the floodgates during a fight.
That's a perfect analogy. iNOS is the body deploying a chemical weapons system during inflammation or infection.
But it's still a reactive free radical. Doesn't it break down quickly?
It does. That reactivity means NO itself only lasts for maybe 4 to 10 seconds in tissue. Very short-lived signal. It breaks down reacting with oxygen to form nitrites, or even faster reacting with hemoglobin and red blood cells to form nitrates, which then get flushed out in urine.
So that short lifespan is protective in a way. But you mentioned free radicals are dangerous. Where's the risk?
The real danger comes when NO bumps into another reactive molecule, specifically superoxide. Superoxide is another type of reactive oxygen species, often produced during metabolism or, significantly, during inflammation.
And NO likes superoxide.
Likes it a little too much. They react incredibly fast to form peroxynitrite.
Peroxynitrite. Sounds ominous.
It is. If NO is the body's precise scalpel, peroxynitrite is, well, you called iNOS a floodgate. Peroxynitrite is more like a bomb. It's a highly potent oxidant.
What does it do?
It causes widespread cellular damage. Lipid peroxidation damages cell membranes, DNA damage, just general cytotoxicity. This is how that beneficial inflammatory NO signal can flip and cause serious tissue injury.
So the body makes this potentially damaging thing when NO meets superoxide. How does it manage that? Is there a defense?
Excellent question. Yes, there is. The body has an enzyme called superoxide dismutase, SOD. Its job is basically to scavenge superoxide radicals, clean them up very quickly.
So SOD removes the superoxide before it can react with NO.
Exactly. By getting rid of the superoxide, SOD prevents the formation of that damaging peroxynitrite. And as a bonus, by removing the molecule that NO reacts with, SOD actually prolongs NO's lifespan slightly and enhances its beneficial actions, like vasodilation.
Clever. So it's not just about making NO, it's also about protecting it and preventing its toxic side reactions.
Precisely. It's all about balance and control. Cellular signaling is as much about cleanup as it is about production.
Okay, that makes sense. Let's pivot to what NO actually does. Its most famous role is in the vascular system, right? As a vasodilator.
Absolutely. It's the key endogenous vasodilator. Made by eNOS in the endothelium, it just diffuses across into the smooth muscle cells lying underneath.
And how does it tell that muscle cell to relax? This is the CGMP pathway, isn't it?
Correct. This pathway is core knowledge. Once NO gets inside the smooth muscle cell, it finds and activates an enzyme called soluble guanylyl cyclase, or SGC.
Activating SGC does what?
It ramps up production of a secondary messenger molecule called cyclic GMP, CGMP. So NO comes in, SGC gets switched on, and suddenly the cell is flooded with CGMP.
Okay, so CGMP is the internal signal. What does CGMP do?
CGMP then activates another player, CGMP-dependent protein kinase, or PKG. Think of PKG as the foreman carrying out CGMP's orders, and PKG basically hits the off switch for muscle contraction in two main ways.
Two ways. Okay, what's the first?
First, PKG directly targets the contractile machinery. It dephosphorylates, meaning it removes a phosphate group from myosin light chain kinase, MLCK. MLCK needs that phosphate to enable actin and myosin to interact and cause contraction. Remove the phosphate, you block the interaction.
So it directly disengages the muscle's motor. What's the second way?
Second, PKG also reduces the amount of calcium available inside the cell. It phosphorylates certain calcium channels in the cell membrane, making it harder for calcium to enter from outside.
And less calcium means?
Less calcium means less activation of MLCK in the first place, because MLCK activation itself depends on calcium binding to calmodulin. So NO via cGMP and PKG both directly inhibits the contractile machinery and reduces the calcium signal needed to turn it on.
Double whammy for relaxation.
Exactly, leading to potent, rapid, smooth muscle relaxation. This is the fundamental mechanism behind how things like acetylcholine and bradykinin cause vasodilation. They trigger NO release, which runs the cGMP pathway.
And when this system fails, if NA production is down.
That's a hallmark of endothelial dysfunction. Losing that basal NO-driven relaxation contributes significantly to the development of high blood pressure, hypertension. It's a key factor.
But it's more than just vessel tone, right? You
Yeah.
mentioned NO protects the vessel itself.
Yes, hugely important. NO is actively anti-atherogenic. It makes the endothelium slippery. It suppresses the ability of white blood cells, leukocytes, to stick to the vessel wall, which is an early step in plaque formation.
Prevents them sticking. What else?
It also inhibits the proliferation, the growth of the smooth muscle cells themselves within the vessel wall. Uncontrolled smooth muscle growth contributes to plaque buildup, so NO actively fights plaque development.
Okay, so wide pipes and clean pipes too. What about blood clotting?
Same pathway, different cell.
Yeah.
NO also activates that cGMP pathway inside platelets. And in platelets, increased cGMP inhibits their aggregation, stops them from clumping together to form a thrombus or clot.
So less NO means stickier platelets, higher risk of clots.
Precisely. Defective endothelial NO function increases the risk of thrombosis. It's really a master regulator, keeps vessels open, prevents plaque, and stops clots. Beautiful system.
Okay, let's switch gears back to that weapon system, NO, the IMS version used in immunity.
Right. So during an infection or severe inflammation, immune cells like macrophages get activated. They switch on INO, producing that flood of NO.
You said NO meets superoxide.
Exactly. Those activated immune cells are also churning out superoxide. NO plus superoxide gives you peroxynitrite. And while peroxynitrite is toxic, it also breaks down into other highly reactive free radicals.
And those radicals are the weapon.
Yes, they have broad antimicrobial action. They can kill bacteria, viruses, fungi, even protozoa. It's a powerful, if somewhat indiscriminate, part of our innate immune defense, sterilizes the area.
But that indiscriminate nature is the problem, isn't it? The double-edged sword.
Absolutely. While NO contributes to useful inflammatory signs, vasodilation causing redness, increased permeability causing swelling, if that iNOS-driven production goes on too long or is too massive, it starts damaging the host's own tissues.
And we see that in chronic diseases.
Definitely. High levels of NO, likely from iNOS, are found in conditions like inflammatory bowel disease, IBD, and asthma, contributing to the ongoing tissue injury.
Which brings us to the critical situation. Septic shock.
Right. Sepsis is basically a body-wide, out-of-control inflammatory response, often to infection. This triggers massive, widespread induction of iNOS in all sorts of cells, immune cells, vessel walls, everywhere.
Leading to
A catastrophic, sustained, overwhelming release of NO throughout the entire circulation.
and the result of flooding the whole body with a potent vasodilator.
Severe refractory hypotension. Blood pressure plummets and becomes incredibly difficult, sometimes impossible to restore, even with potent vasopressor drugs. This leads to organ failure because tissues aren't getting enough blood flow.
It seems logical then. If excess NO is causing the life-threatening low blood pressure in sepsis, shouldn't we block NO production, use NOS inhibitors?
You'd think so. It was a major hope. But clinical trials using various NOS inhibitors in septic shock have been largely disappointing.
Why? What happened?
They often showed no survival benefit, and sometimes patients did worse. The thinking now is that while blocking the excessive iNOS might seem good, these inhibitors often also block the essential life-sustaining NO production from eNOS.
Ah, the NO needed for normal vessel function, kidney blood flow, heart function.
Exactly. You might be shutting down the pathological NO, but also the vital physiological NO. It highlights just how tricky it is to intervene in these complex systems. Blocking one part can have unintended harmful consequences elsewhere.
Fascinating challenge. Okay, what about NO's role in the nervous system? It's not just blood vessels and immunity.
Not at all. NO acts as a neurotransmitter, or perhaps more accurately, a neuromodulator in the brain and peripheral nerves. Both NAOS and even eNOS are found in neurons.
How is it released? Is it like standard neurotransmitters?
Completely different. This is unique. ONO isn't stored in little packages, in vesicles like acetylcholine or dopamine, and it's not released by that standard process of exocytosis.
So how does it get out?
It's made on demand. When NOS is activated, usually by calcium influx, it instantly synthesizes NO, and the NO simply diffuses. Straight out of the neuron, across the synapse, even through neighboring cells.
Wow. No storage, just make and release by diffusion. Very efficient, very rapid.
And very short range. It's prominent in neurons that use glutamate, especially involving the NMDA receptor. When NMDA receptors are strongly activated, they let in a lot of calcium.
Which activates NNOS, releasing NO.
Precisely. And this NO release is involved in some really fundamental brain functions. It modulates nociception, our perception of pain. It helps regulate local blood flow within the brain. And it's critically involved in learning and memory formation.
Learning and memory.
Wow. Through a process called synaptic plasticity, the ability of connections between neurons to strengthen or weaken over time. NO is thought to be a key player here, potentially acting as a retrograde messenger.
Retrograde, meaning it goes backwards.
Yes. It might diffuse from the postsynaptic neuron, the one receiving the signal, back to the presynaptic neuron, the one sending the signal.
You tell the sending neuron.
Something like, okay, I got that signal loud and clear, send more next time. Or modulate the release somehow. It's a way for the receiving cell to influence the sender, strengthening that particular connection.
That's incredibly sophisticated. But just like in inflammation, can too much NO in the brain be bad?
Absolutely. Excessive NO production, often linked to overactivation of those glutamate receptors, something called excitotoxicity, is implicated in neuronal damage after strokes, for example. And there are links between aberrant NO signaling and neurodegenerative diseases like Huntington's, Parkinson's, maybe ALS too.
Okay. What about nerves outside the brain? Peripheral roles.
The most famous one is penile erection. NO is the key mediator here. It's released from specific nerves in the penis non-adrenergic, non-cholinergic, or NANC nerves.
And it does the same thing, relaxes smooth muscle.
Exactly. It relaxes the smooth muscle in the corpora cavernosa, the spongy tissue, allowing blood to flow in and cause tumescence.
Which brings us neatly to Viagra.
Super nicely. Sildenafil, Viagra, and similar drugs are PDE5 inhibitors. Phosphodiesterase type 5 is the enzyme that specifically breaks down cGMP in these tissues.
So by blocking the breakdown enzyme,
you make the cGMP, produced in response to NO, last much longer. You amplify and prolong NO's natural relaxing effect. A brilliant pharmacological strategy based directly on understanding this NO-cGMP pathway.
amazing. Any other peripheral roles?
It seems NO also helps relax the pyloric sphincter, the muscle-controlling passage from the stomach to the intestine. Problems with NO signaling there might be involved in conditions like pyloric stenosis in infants.
Okay, given how important NO is for relaxation, it makes sense we'd want drugs that mimic it. The NO donors.
Exactly. Nitrodilators, or NO donors. These are compounds that, through various mechanisms, release NO or something chemically similar that can activate the same pathway in smooth muscle.
The classic is glycerol trinitrate, GTN nitroglycerin, used for angina for over a century.
The absolute prototype. It's very effective for rapid relief of angina pain. Its main action is dilating veins more than arteries.
Veins. Why veins predominantly?
It reduces the amount of blood returning to the heart, reduces preload. This decreases the heart's workload and oxygen demand. It also dilates coronary arteries, improving oxygen supply. The reason for the venous preference seems to be the enzyme needed to release NO from GTN.
Which enzyme is that?
It's a mitochondrial enzyme, aldehyde dehydrogenase type 2, ALDH2. And it turns out this enzyme is much more abundant in venous smooth muscle than arterial. Hence, veins are better at activating GTN.
Clever.
Yeah.
But GTN has that notorious problem. Tolerance.
Yes, a major clinical issue. If you use GTN continuously, like with patches, its effect rapidly diminishes. Tolerance develops quickly. The leading theory involves those reactive oxygen species again.
How so?
The process of metabolizing GTN might generate ROS, which then damage or inhibit that crucial mitochondrial ALDH2 enzyme. The cell essentially becomes unable to process the GTN to release NO.
And you mentioned something important earlier. GTN doesn't really affect platelets, unlike native NO.
Correct. Organic nitrates like GTN generally don't inhibit platelet aggregation significantly. Platelets seem to lack the necessary enzyme machinery, like ALDH2, to effectively generate NO from these drugs. A key difference to remember compared to endogenous NO.
Okay, so GTN, venous dilation, rapid relief, but tolerance. What's the other big NO donor?
Sodium nitroprusside. This one's given intravenously, usually in hypertensive emergencies or during surgery for controlled hypotension.
How is it different from GTN?
It's a much more balanced vasodilator. It dilates both arterioles and veins pretty equally. This leads to a potent reduction in both preload and afterload, dropping blood pressure very effectively. It seems to release NO more directly, maybe non-enzymatically, and also directly activates SGC.
Does it have the tolerance issue?
No, that's a major advantage. Tolerance is virtually absent with nitroprusside, even with prolonged infusions. It remains effective.
But there's a catch, right? Cyanide.
Yes, that's the significant risk. When nitroprusside breaks down, it releases not only NO, but also 5 cyanide ions per molecule.
Cyanide. That's toxic.
Highly toxic. The body has a mechanism to detoxify it, converting it to thiocyanate using sulfur donors, mainly in the liver. But this process can be overwhelmed, especially with high doses or prolonged infusions or in patients with liver disease.
And thiocyanate can build up too.
Yes, thiocyanate is less toxic than cyanide, but can still cause problems if it accumulates, like confusion, psychosis, especially in patients with kidney failure, as it's cleared by the kidneys. So nitroprusside requires very careful monitoring.
Okay, one more therapeutic use. Inhaled NO gas itself.
Right. Using the actual gas, NO, delivered via inhalation. This is specifically used in newborns with severe respiratory distress who also have pulmonary arterial hypertension, PH, dangerously high pressure in their lung arteries.
How does inhaling it help?
Because it's inhaled, the NO gas preferentially goes to the parts of the lung that are well ventilated, the parts getting oxygen. It then dilates the blood vessels only in those well ventilated areas.
So it improves the matching of ventilation, air, and perfusion.
Precisely. It redirects blood flow away from poorly ventilated lung areas towards the areas where oxygen exchange can actually happen. This improves oxygenation and simultaneously lowers the overall pressure in the pulmonary artery. It's a very targeted, very clever therapy for a critical condition.
Okay, that covers NO pretty thoroughly. Now for the complete shift to the vasoactive peptides. The complex messengers.
Yes. Moving from the simple gas to these intricate amino acid chains, they generally act via specific receptors, often G-protein coupled receptors, and have much more complex synthesis and regulation. Let's start with the constrictors.
And the king of the constrictors is?
Endothelin, specifically endothelin-1 or ET1. There are actually 3 related peptides, ET1, ET2, and ET3, all 21 amino acids long. But ET1, produced mainly by the vascular endothelium, is the big player. It's arguably the most potent vasoconstrictor substance the body makes.
How is it made? Is it complex like the NO's pathway?
It's a multi-step process. It starts as a large precursor protein, prepro ET1. This gets chopped down to an intermediate called big ET1, which has 38 amino acids and is inactive.
Then the final step.
The final step is cleavage by a very specific enzyme called endothelin converting enzyme, ECE. ECE cuts big ET1 down to the active 21 amino acid ET1 peptide. ECE is a key control point.
And what controls the production of ET1? Is it regulated like NOS?
It's heavily regulated, yes, often by factors involved in stress or injury. Things that increase ET1 synthesis include growth factors like TGF-beta, inflammatory cytokines like IL-1, and other vasoconstrictors like angiotensin II and vasopressin.
So vasoconstrictors promote the production of another vasoconstrictor, a positive feedback loop.
In a way, yes, amplifying the constrictor response. But importantly, ET1 production is suppressed by the vasodilating signals we just talked about, and another vasodilator called PGI2, prostacyclin, and the natriuretic peptides we'll discuss later.
The push and pull again. Constrictors up ET1, dilators down ET1.
Exactly, homeostasis in action.
What does ET1 actually do? Just constrict blood vessels?
It causes profound and very long-lasting vasoconstriction. It's particularly potent in the arteries of the lungs, kidneys, and heart. Interestingly, if you inject ET1, you see a weird initial effect.
What's that?
A brief, transient drop in blood pressure.
A drop? From a potent vasoconstrictor. Why?
Because ET1 can also stimulate the release of NO and PGI2 from the endothelial cells themselves, probably via the ETB receptor we'll mention. So you get a fleeting vasodilation before the main powerful direct constrictor effect on the smooth muscle kicks in.
Okay, so brief dip, then a sustained major rise in blood pressure. Increases heart rate too.
Yes, it increases heart rate and contractility. But ET1 does more than just squeeze vessels. It has crucial long-term effects. It's strongly mitogenic.
Mitogenic meaning?
It promotes cell growth and proliferation. Specifically, it encourages the growth of vascular smooth muscle cells, cardiac muscle cells, myocytes, and fibroblasts, cells that produce connective tissue.
So in the long run, too much ET1 doesn't just constrict vessels, it helps remodel them, thickens them, contributes to fibrosis in the heart.
Precisely. This mitogenic action is why chronic overproduction of ET1 is heavily implicated in the pathology of diseases involving tissue remodeling, like pulmonary arterial hypertension, systemic hypertension, atherosclerosis, and chronic heart failure. It's a driver of structural damage.
How does it signal? What are the receptors?
There are 2 main receptor subtypes, both GPCRs, ETA and ETB.
ETA does what?
The ETA receptor is found mainly on vascular smooth muscle cells. It has a higher affinity for ET1 than for ET2 or ET3. When ET1 binds to ETA, it activates the GQ protein pathway, stimulating phospholipase C, generating IP3 and DEG, leading to calcium release from internal stores and ultimately strong muscle contraction. This is the main vasoconstrictor pathway.
And ETB?
The ETB receptor is a bit more complex. It binds all three endothelins, ET1, NOTORS2, NOTORS3, with roughly equal affinity. You find ETB receptors on smooth muscle, too, where they can also cause contraction. But importantly, ETB receptors are also found on the endothelial cells.
And what do the endothelial ETB receptors do?
When activated, they trigger the release of those vasodilators, NO and PGI2. This is the mechanism behind that initial transient drop in blood pressure we mentioned. It's a kind of counter-regulatory effect. ETB receptors also play a role in clearing ET1 from the circulation.
So ETA is mainly constriction and proliferation, while ETB is some constriction, but also vasodilation via NO-PGI2 release and clearance. But overall, excess endothelin is bad news.
Very bad news, especially in conditions like PAH.
Right, which leads us directly into the drugs designed to block these receptors, the endothelin receptor antagonists, or ERAs.
Correct. And their only approved clinical indication currently is pulmonary arterial hypertension, PAH. This is a devastating disease where the pulmonary arteries become thickened and constricted, leading to extremely high pressure in the lungs, often defined as mean pulmonary artery pressure over 25 mmHg at rest.
And endothelin, particularly via ETA receptors, is a key driver of that remodeling and constriction in PAH.
Absolutely. ETA receptors are significantly upregulated in the pulmonary arteries of PAH patients, so blocking this pathway makes perfect sense. ERAs, along with drugs that boost NO or PGI2 signaling, are the cornerstones of PAH therapy, though unfortunately they are palliative, not curative.
Let's talk about specific ERA drugs. There are three main ones approved.
Yes. The first one approved was bosentan. It's a dual antagonist, meaning it blocks both ETA and ETB receptors.
Blocking both.
Yeah.
Any downsides?
The main issue with bosentan is its metabolism and potential toxicity. It's broken down by liver enzymes, CYP2C9 and CYP3A4. This leads to two problems. First, a risk of dose-dependent liver injury, hepatotoxicity. Patients on bosentan require mandatory monthly liver function tests.
Monthly tests.
Okay.
What's the second problem?
Because it's metabolized by those CYPs and it also induces them, it has significant drug interactions. Most notably, it can reduce the effectiveness of drugs like warfarin, a blood thinner, and oral contraceptives. Big clinical considerations.
So liver monitoring and drug interactions. That led to newer agents.
Exactly. The next major one was ambrisentan. This one is selective for the ETA receptor.
Selective ETA. Does that help with the side effects?
It seems to. Ambrisentan has a significantly lower risk of causing liver enzyme elevations compared to bosentan. The need for routine monthly liver monitoring is less stringent, though still recommended initially. It also has a nice long half-life, around 12-15 hours, allowing for once-daily dosing.
Fewer drug interactions, too.
Generally lower potential for significant interactions compared to bosentan, and ambrisentan was part of a landmark trial, the AMBITION study.
What did AMBITION show?
It showed that starting patients with PAH on combination therapy upfront, ambrisentan blocking ET, plus tadalafil, a PDE5 inhibitor boosting NO's effect, was significantly better than starting with just one drug. It improved outcomes and reduced the risk of clinical worsening.
Really highlights that strategy of hitting both the constrictor and dilator pathways simultaneously.
Precisely. Block the bad, ET, boost the good, NO-cGMP.
Okay, so bosentan, dual liver risk, ambrisentan, selective ETA, less liver risk. What's the third one?
The newest is macitentan. This is another dual ETA-B antagonist, but it was designed for improved properties.
Like what?
It has very high affinity, especially for ETA, and it binds really tightly, producing what's called an unsurmountable blockade. This means even if ET1 levels go up, they can't easily knock macitentan off the receptor. It also has a long half-life, about 17 hours, plus an active metabolite that extends its action.
Did it show better outcomes?
Yes, in its major clinical trial, SERAPHIN, macitentan significantly reduced the risk of death or major PAH-related morbidity events by about 45% compared to placebo. A real advance.
Are there common side effects across all these ERAs?
Yeah.
Besides the liver issue with bosentan?
Yes, because they are fundamentally vasodilators. By blocking the constrictor ET1, they can cause typical vasodilator side effects: headache, flushing, and peripheral edema swelling, usually in the ankles.
Edema.
It's partly due to the vasodilation itself altering fluid dynamics, and partly due to some fluid retention. This can also lead to a mild hemodilution, meaning the blood is slightly more watery, which shows up as a small drop in hemoglobin levels. A mild anemia is common.
Okay, but there's one overriding critical warning for all ERAs, isn't there?
Absolutely crucial, teratogenicity. All ERAs have shown significant risks of causing birth defects in animal studies. They are absolutely contraindicated in pregnancy.
Meaning women who could become pregnant need extremely strict precautions.
Yes. Mandatory pregnancy testing before starting, monthly during treatment, and for a month after stopping. Use of reliable contraception is essential. There are strict prescribing programs like REMS in the U.S. to manage this risk. It cannot be overstated.
Understood. Very serious risk. Okay, let's touch on a couple of other vasoconstrictor peptides before we move to the dilators. Neuropeptide Y.
Neuropeptide Y, NPY. This is one of the most abundant neuropeptides in the entire nervous system, central and peripheral. Its key location peripherally is alongside noradrenaline, norepinephrine, in sympathetic nerve terminals.
So it works with noradrenaline, a co-transmitter.
Exactly. It amplifies and modulates the sympathetic response. At the target tissue, like blood vessels, it has two main effects. First, it can cause a direct, slow, long-lasting contraction itself. Second, and maybe more importantly, it potentiates the vasoconstriction caused by noradrenaline, makes NA work better.
So it strengthens the fight-or-flight vascular response. What about on the nerve ending itself?
Projunctionally, it often acts as a brake. It can inhibit the release of various neurotransmitters, including noradrenaline itself, but also acetylcholine, dopamine, serotonin, glutamate. It's a modulator.
And in the brain. NPY does a lot there, too.
Huge role centrally. It's one of the most powerful stimulators of feeding and appetite known, deeply involved in regulating energy balance, stress responses, anxiety, circadian rhythms. Very complex.
Are there different NPY receptors for these roles?
Yes, several subtypes. The main one driving vasoconstriction peripherally is the Y1 receptor. The Y2 receptor is often involved in that projunctional inhibition. And the Y5 receptor, found mainly in the brain, is heavily linked to controlling food intake and the stress axis, HPA axis.
Have we tried to make drugs targeting these, like Y1 blockers for hypertension?
Y1 antagonists were developed. They help show that NPY probably doesn't play a major role in regulating normal blood pressure, but maybe more in stress-induced hypertension. The big interest, though, was Y1 and especially Y5 antagonists as potential anti-obesity drugs.
Yeah, any success there?
A lot of research, but so far, no NPY antagonists have made it to market as approved drugs for obesity or anything else.
Okay, one more constrictor, urotensin 2I.
Urotensin 6, UII. A smaller cyclic peptide, 11 amino acids. It's a very potent constrictor of arteries, particularly vascular smooth muscle. Can also depress heart function. Signals through its own GBCR, the UT receptor, which uses the GQ pathway like ETA.
Does it have a normal physiological role?
That's unclear. Like NPY, it doesn't seem essential for normal blood pressure control. However, levels of urotensin II are found to be elevated in several disease states: hypertension, advanced heart failure, diabetic kidney disease.
Suggesting it might contribute to the pathology in those conditions.
That's the idea. It might become important once disease is established. Antagonists have been tested, for example, in diabetic nephropathy and even asthma, based on some smooth muscle effects. But again, no drugs targeting urotensin II have been successfully developed yet.
Okay, enough constrictors. Let's talk about the peptides that dilate vessels and modulate things, the counterplayers.
Yes, let's shift to the vasodilators. And a really potent, historically important group are the kinins, primarily bradykinin, and a related peptide, calodin.
How are these made? Are they always floating around?
No, they're generated on demand, usually in response to tissue injury or inflammation. There are precursor proteins in the plasma called kininogens, types of globulins. Enzymes called kallikreins cleave these kininogens to release the active bradykinin or calodin peptides.
So, injury triggers kallikreins, which cut kininogens, releasing kinins.
That's the basic cascade. And these kinins are incredibly potent, but also incredibly short-lived. Their half-life in blood is less than a minute.
Wow, less than a minute. What breaks them down so fast?
Primarily an enzyme called kininase II. It rapidly inactivates them. And here is perhaps the single most clinically relevant connection for this entire group.
Okay.
Kininase II is exactly the same enzyme as angiotensin-converting enzyme, ACE.
Wait, ACE, the enzyme that makes the angiotensin II, the vasoconstrictor,
Yeah.
that same enzyme destroys bradykinin, the vasodilator.
Precisely. ACE has a dual role. It promotes the RAS system, making ANG2, and simultaneously shuts down the opposing kinin system, destroying bradykinin. This is fundamentally important for understanding ACE inhibitor drugs.
We'll come back to that. What does bradykinin actually do, besides vasodilate?
It's an extremely potent vasodilator, even more so than acetylcholine or histamine. It works mainly by stimulating endothelial cells to release our old friends, NO and PGI2.
So it uses the NO pathway. What else?
It dramatically increases capillary permeability, makes blood vessels leaky. This causes fluid to escape into tissues, leading to swelling, edema, the classic wheel and flare reaction you see in skin tests. And crucially, it's a powerful activator of pain nerves.
Pain.
Yes, it strongly stimulates nociceptive afferent nerve endings, causing that characteristic burning sensation associated with inflammation. In fact, bradykinin is sometimes used experimentally to induce pain when testing new analgesics. It's a key mediator of inflammatory pain.
So vasodilation, permeability, pain, classic signs of inflammation.
Absolutely. Bradykinin is a major player in producing all the cardinal signs of inflammation. There are two main bradykinin receptors, B1 and B2.
What's the difference?
The B2 receptor is the main one. It's constitutively expressed, meaning it's always present on cells like endothelium and neurons. It mediates most of the acute effects we just discussed: vasodilation, increased permeability, acute pain.
And the B1 receptor?
The B1 receptor is different. It's normally present at very low levels, but its expression is dramatically induced by inflammation, by cytokines like IL-1 and TNF-alpha. Once B1 is upregulated, it seems to contribute more to chronic inflammatory processes and chronic pain states. It also stimulates further release of inflammatory mediators.
So B2 for acute effects, B1 ramps up during chronic inflammation. Interesting distinction. Is there a disease directly caused by too much bradykinin?
Yes, a rare but potentially fatal genetic disorder called hereditary angioedema, HAE.
Angioedema, that's severe swelling, right?
Yes, massive, unpredictable swelling attacks that can affect the face, limbs, gut, and most dangerously, the airway. HAE is usually caused by a deficiency or dysfunction of a protein called C1 esterase inhibitor.
And C1 esterase inhibitor normally does what?
It normally acts as a brake on the kallikrein system, preventing excessive kinin generation. Without that brake in HAE, patients produce way too much bradykinin, leading to these severe swelling episodes.
How do you treat an acute HAE attack?
You need to block the bradykinin directly. The best current treatment is a drug called icatibant. It's a synthetic peptide that acts as a selective B2 receptor antagonist. It blocks bradykinin from activating its main receptor.
And it works well.
Yes, it's effective in terminating acute HAE attacks. And interestingly, icatibant has also shown effectiveness in treating the angioedema that can be a rare but serious side effect of
ACE inhibitors.
Yeah,
Because they block kininase II, letting bradykinin build up.
exactly. The fact that a bradykinin B2 blocker treats ACE inhibitor-induced angioedema is the definitive proof that this side effect is mediated by excess bradykinin accumulation. It beautifully ties the kinin system and ACE inhibitors together.
Fantastic connection. Okay, moving on to another major vasodilator peptide system, the natriuretic peptides?
Right, the NTs. These are the body's natural defense against volume overload and high pressure. There are three main members: atrial natriuretic peptide, ANP, brain natriuretic peptide, BNP, and C-type natriuretic peptide, CNP.
ANP from the atria, BNP originally found in brain, but mostly from ventricles.
Yeah.
What triggers their release?
The primary stimulus is mechanical stretch. When the atria or ventricles of the heart are stretched, usually due to increased blood volume or pressure, they release ANP and BNP respectively.
So they're released when the heart is working too hard, essentially.
That's a good way to put it. And BNP in particular has become a crucial clinical biomarker. Measuring BNP levels in the blood is a standard way to help diagnose, monitor the severity, and guide treatment of chronic heart failure, CHF. Higher BNP means more ventricular stretch, worse heart failure.
What do these NPs do once released? They're natriuretic, meaning.
Natriuretic means causing sodium Na plus excretion in the urine. Diuretic means causing water excretion. NPs do both. They are fundamentally counter-regulatory to the renin-angiotensin-aldosterone system, RAS.
They oppose RAS.
How? In multiple ways. Vasculature. They cause systemic vasodilation, both arteries and veins, which reduces the workload on the heart, reduces afterload and preload, kidneys. They increase renal blood flow and the glomerular filtration rate, GFR, while directly inhibiting sodium reabsorption in the kidney tubules. This promotes salt and water loss.
So they lower blood volume and pressure. Do they affect RAS directly?
Yes. They also inhibit the release of renin from the kidneys. Less renin means less angiotensin II production, and consequently less aldosterone release. So they shut down the whole RAS cascade. It's a beautiful counterbalancing system.
How do they signal? What are their receptors?
ANP and BNP primarily act on receptors called NPRA or ANPA and NPRB or ANPB. These are not typical GPCRs. They have intrinsic guanylyl cyclase activity.
Like the soluble GC that NO activates.
Similar end result, but these are membrane-bound enzymes linked directly to the receptor. Binding of ANP or BNP activates the guanylyl cyclase domain inside the cell, leading to increased production of cGMP.
Ah, cGMP again, leading to relaxation.
Exactly. Same second messenger pathway as NO, leading to smooth muscle relaxation and other effects. There's also a third receptor, NPRC or AMPC, which doesn't have GC activity. Its main role seems to be clearing NPs from the circulation, acting like a clearance receptor to terminate the signal.
Okay, so MPs are protective. They lower volume and pressure via cGMP. If they're so good, can we boost their effects pharmacologically?
That was the next logical step. Since NPs are beneficial, maybe we can stop their breakdown. There's an enzyme called neprilysin, or neutral endopeptidase, NEP, that is a major route for degrading NPs.
So inhibit neprilysin, NPs stick around longer.
That's the idea. Neprilysin also breaks down other vasodilator peptides, including bradykinin, by the way. So inhibiting it should boost multiple beneficial systems.
Did it work?
Yes, this led to the development of sacubitril. It's an orally active prodrug that gets converted to a potent neprilysin inhibitor. However, just inhibiting neprilysin alone wasn't enough, likely because the body compensates via RAS.
So you need to block RAS too.
Exactly. The breakthrough came from combining the neprilysin inhibitor sacubitril with an angiotensin receptor blocker, ARB, specifically valsartan. This combination drug is known as an ARNI angiotensin receptor neprilysin inhibitor.
Sacubitril valsartan. I've heard of that. Entresto is a brand name. Big deal in heart failure.
It's a huge deal. The PARADIGM-HF trial was a landmark study. It compared sacubitril valsartan against the standard ACE inhibitor enalapril in patients with advanced chronic heart failure, class 2.
And the result?
The results were stunning. Sacubitril valsartan was significantly superior to enalapril. It resulted in about a 20% greater reduction in the combined endpoint of cardiovascular death or hospitalization for heart failure.
20% better than the gold standard. That's massive.
It truly revolutionized heart failure management. ARNIs are now a cornerstone therapy, often preferred over ACE inhibitors or ARBs alone in appropriate patients with symptomatic heart failure. It validates the strategy of simultaneously boosting the protective NP system while blocking the detrimental RAS system.
Incredible synergy. Okay, let's move to the final group, tachykinins and CGRP, pain and inflammation again.
Yes, these are heavily involved in sensory nerve signaling, inflammation, and pain. The main tachykinin in mammals is substance P.
Substance P. What does it do to blood vessels?
It's complicated. It's a potent arterial dilator, but it achieves this indirectly by acting on endothelial cells via NK1 receptors to release NO. However, it directly constricts veins and also causes contraction in many non-vascular smooth muscles, like in the gut or airways, hence the name tachykinin rapid contraction.
Where is SP clinically important?
Its most well-established clinical role is in emesis, nausea, and vomiting.
How is substance P involved in vomiting?
It's released from nerve endings in the gut, especially in response to noxious stimuli like chemotherapy drugs or postoperative irritation. This SP travels via the blood and also signals within the brainstem, specifically in the chemoreceptor trigger zone, CTZ, and the nucleus tractus solitarius, NTS, which coordinate the vomiting reflex.
So blocking SP should prevent vomiting.
Exactly. Drugs that block the main SP receptor, the NK1 receptor, are effective antiemetics. Aprepitant and its IV prodrug fosaprepitant are NK1 antagonists widely used, often in combination with other antiemetics, to prevent delayed nausea and vomiting caused by highly immunogenic chemotherapy, and also for postoperative nausea and vomiting, PONV.
Okay, so SPNK1 in emesis. What about CGRP? Calcitonin gene-related peptide.
CGRP is another peptide found alongside substance P in many sensory nerves. It's an extremely potent vasodilator, maybe even more potent than SP, but its mechanism is different.
Different how? Not NO.
No, CGRP doesn't rely on NO release. It acts directly on smooth muscle via its own receptors, which are coupled to adenyl cyclase, AC. This leads to an increase in cyclic AMP, CAMP, which causes relaxation. So CGRP uses the KAMP pathway, whereas NO and NPs use the CGMP pathway.
CMP, okay. Where is CGRP clinically relevant? This is the migraine story, right?
This is the migraine story. It's been a huge area of research and breakthrough.
Yeah.
CGRP plays a central role in migraine pathophysiology. Sensory nerves that innervate the meninges, the coverings of the brain, specifically nerves from the trigeminal ganglion, release large amounts of CGRP during during a migraine attack or possibly just before it starts.
And this release CGRP does what?
It causes pronounced vasodilation of the meningeal blood vessels and also promotes what's called neurogenic inflammation release of inflammatory mediators from the nerves themselves. This combination of vasodilation and inflammation is thought to be a key trigger or component of the migraine pain cascade. CGRP levels are measurably elevated in people during a migraine attack.
So the logical step was to block CGRP.
Precisely, and this led to one of the biggest advances in neurology in decades. Several drugs have been developed to target the CGRP pathway, most notably monoclonal antibodies.
Antibodies against CGRP or its receptor.
Both strategies exist. For example, aranumab is a fully human monoclonal antibody that blocks the CGRP receptor. Others directly bind and neutralize the CGRP peptide itself. These are given by injection, usually monthly or quarterly.
And they work.
They are remarkably effective for many patients for migraine prophylaxis, preventing attacks. Clinical trials showed they can significantly reduce the number of migraine days per month, often cutting them by half or more for responders. It's been life-changing for many people with chronic or frequent episodic migraine.
A true targeted therapy based on understanding the peptide's role. Amazing. Any other vasodilator peptides we should briefly mention?
Just quickly, two others are vasoactive intestinal peptide, VIP, and adrenomedulin, AM.
VIP.
VIP is often found as a co-transmitter with acetylcholine, particularly in parasympathetic nerves. It's a potent vasodilator, also relaxes smooth muscle in the bronchi and gut, and increases heart contractility. It acts via VPAC receptors linked to the Gs and CANDMP production, mostly acts locally, paracrine signaling.
And adrenomedulin.
AM belongs to the calcitonin family, like CGRP. It causes long-lasting vasodilation and hypotension. Its receptor is structurally related to the CGRP receptor. Levels of AM are often elevated in conditions like hypertension and heart failure, suggesting it might be part of a compensatory response trying to counteract the vasoconstriction.
But no drugs targeting VIP or AM yet.
But not in widespread clinical use, no. Their roles are still being fully elucidated, and targeting them pharmacologically has proven complex.
Okay, we've covered a huge amount from the simplest gas to these complex peptide families. Let's try to synthesize this.
Absolutely. If we connect this to the bigger picture, what we've seen is this incredible biological balancing act. You have these powerful constrictor systems, endothelin, NTY, often working alongside the renin-angiotensin system, driving pressure up, sometimes causing remodeling.
When opposing them, you have the dilator and volume-lowering systems,
Right?
the rapid versatile gas NO, the potent inflammatory kinins like brodykinin, the volume-sensing natriuretic peptides, and potent sensory nerve dilators like CGRP.
It's a constant push and pull, maintaining homeostasis. And when that balance gets severely disrupted in disease, think PAH with too much endothelin, or heart failure where RAS is overactive and NPs are trying to compensate, that's where our understanding allows for targeted intervention.
And the level of sophistication in targeting is really impressive now. It's moved beyond just general vasodilators.
Completely. We're talking about highly specific endothelin receptor antagonists for PAH, neprilysin inhibitors carefully designed to boost endogenous NPs while blocking RAS and heart failure. Monoclonal antibodies precisely targeting CGRP for migraine. These are elite clinical tools born directly from mastering this fundamental peptide and gas signaling.
It really is astonishing how this dialogue between a tiny gas molecule that lasts seconds and these elaborate peptide chains governs so much of our cardiovascular health, nerve function, and response to injury.
It's the elegance of physiology. So here's something for you, the listener, to mull over, connecting some threads. We know endogenous vasodilators like NO, kinins, and NPs are generally protective. We know ACE inhibitors, a cornerstone of treating hypertension and heart failure, work partly by blocking the breakdown of bradykinin via kinase II.
But that same mechanism causes the potentially life-threatening side effect of angioedema in some people due to bradykinin buildup.
Exactly. So the provocative thought is, how might future drug development build on the success of ARNIs, blocking RAS plus boosting NPs, to further harness the protective kinin system, perhaps by finding ways to enhance its benefits while completely designing out the dangerous angioedema risk? Can we find a way to get the good kinin effects without the bad? That's the kind of challenge that tackling this fundamental science prepares us for.